Hello everyone, I'm Dr. Arpita. A very warm welcome to all of you. Today we are going to solve KSET 2020 Life Science question paper. So let's get started. This is the first part of our lecture. It has 50 questions. In the second part, we'll do the rest of the questions. So here the first question says, the constituent monosaccharide in chitin is. 4 options are given. A, D-glucose, option B, D-xylose, option C, N-acetyl D-glucosamine, and option D, D-galactose. We know chitin is the chief component of fungal cell wall, and also it is found in insects. So here the chief component is N-acetyl D-glucosamine. Coming to the second question, an example of aromatic acid is. Here it is a very easy question. We can all see that methionine, valine, alanine are not aromatic. The only option here is option D, that is tyrosine. Coming to question number 3, which functional group is never found in alpha amino acids? CHO group is never found in alpha amino acids. So our right answer is C, that is CHO. Question number 4. Ramachandran plot displays option A, allowed angles of phi and psi for polypeptide backbone. Option B, preferred amino acids in a helix. Option C, the hydropathy of amino acids, and option D, angles of rotation of R group of amino acid. So we should just once recall what is Ramachandran plot. We had talked about Ramachandran plot when we were studying about amino acids. So what does Ramachandran plot tell us? Ramachandran plot shows the statistical distribution of the combinations of the backbone dihedral angles phi and psi. It actually shows us the allowed regions of, in theory, the allowed regions of the Ramachandran plot show which values of phi and psi angles are possible for an amino acid. Say X in a, say alanine, X alanine tripeptide. So this is what is Ramachandran plot telling us. So our correct answer is option A, allowed angles of phi and psi for polypeptide backbone. Coming to question number 5. Left-handed helix is found in. It is found in Z DNA. From the other options we can see B DNA, A DNA, C DNA. These are all right-handed helix. So we are only left with one option, option D, that is Z DNA. Question number 6. pH is equal to pK when option A, proton acceptor is equal to half of proton donor. B, proton acceptor is equal to proton donor. Option C, proton acceptor is equal to twice proton donor, and option D is proton twice proton acceptor is equal to proton donor. Actually option D and option A are same. If you see it, if you observe it nicely, you'll see option A and option D are same. Our correct answer is, see, A, C and D can never be possible. pH and pK, when they are equal, it can only happen when proton acceptor and proton donor are equal. So our correct answer is option B. Now coming to question number 7, match the following with reference to protein structure and modifying agents. So here two columns are given. Column A has names of certain modifying agents, and column B has the protein structures. So the 4 components in column A are mercaptoethanol, guanidine hydrochloride, urea and dithiotritol. Column B has polypeptide bond, peptide bond, disulfide bridge, hydrogen bond and ionic bond. So let's start matching them. We know mercaptoethanol, or say beta mercaptoethanol, is a reagent that breaks disulfide bonds in a protein. So option 1 goes with B. So we'll have to see which option among A, B, C, D has 1 associated with B. We'll choose that, and then we'll also tally the others as well, and this is how we will eliminate the rest of the option. Here we can see only option B has 1 associated with B. So even if you don't know the rest of the option, here you can easily make a correct choice. So beta ethanol or mercaptoethanol is matched with disulfide bond. Guanidine hydrochloride is matched with hydrogen bond. Urea is matched also with hydrogen bond, and the last one, dithiotritol, is matched with disulfide bridge. So our correct answer is option B. Coming to the next question, that is question number 8. Question number 8 says match the column. Again you have matched the column. Here column A gives you the names of the vitamins, the name with letter code and their chemical names. Vitamin D, E, B6, B1, and here in column B you have tocopherol, thiamine, pyridoxine and calciferol. So here let's see which one is matched with which one. Vitamin D is known as calciferol. Vitamin E is known as tocopherol. Vitamin B6 is known as pyridoxine and B1 is known as thiamine. So this goes with, or this matches with option C. Coming to question number 9, what are the ion carriers located in a cell? In the cell, ion carriers are only the cell membrane among these given options. Nucleus doesn't carry any ion, cell wall never, intracellular space also doesn't carry. So what carries, what has the ion carriers are cell membrane. Intercellular space you don't have any ion carriers, in the cell wall you don't have, in the nucleus you don't have. So you're left with only one answer, that is cell membrane. Coming to question number 10, which of the following cell organelle contain DNA? Golgi has no DNA of its own, ER has no DNA, ribosome has no DNA, only mitochondria. Had there been another option, chloroplast, we would have said chloroplast and mitochondria. But among these four, it's only mitochondria. That is why we call it semi-autonomous organelles. Next, in which of the following organelles beta oxidation of long chain fatty acids is initiated with an object of shortening the chain length? Option A, lysosomes, peroxisomes, microsomes and Golgi bodies or Golgi apparatus. Correct answer is peroxisomes. Question number 12. The cell division, which consists of nuclear division, that is mitosis, followed by cytoplasmic division, that is cytokinesis, occurs during G1S, G2M. Which of the phases? M stands for mitosis. So, it's very much evident that the correct answer is M. G1 and G2 are not your dividing, doesn't include the dividing phase, and S is the phase where replication of DNA occurs. Nuclear division doesn't occur in that phase. Next, coming to question number 13. Which of the following pair matches with membrane transport process and its primary function? Exocytosis, the movement of macromolecules into the cell. Very much wrong. Exo means outside. So, that's wrong. Pinocytosis, the uptake of water and solutes into the cell. No. Osmosis, passive diffusion of small solutes. Correct. Phagocytosis, secretion of large particles from the cell. No. So, our correct answer is C. Coming to 14, the core of nucleosome consists of H1, H2A, H2B, H4. No. H2A, H2B, H3, H4. Correct. So, option B is your correct answer. C and D also is not matching. So, option B is your correct answer. Coming to question number 15, the concept of pH hemostasis is the ability of the microbe to control its internal pH. Absolutely correct. The ability of microbe to control its external pH. How can the microbe control the pH of the external environment? It is so difficult and it's not possible also. So, this is wrong. The ability of the microbe to grow on acidic pH, not at all. Grow on alkaline pH, that is also baseless. So, our correct answer is A. Next question. A metabolic pathway that involves part of the Krebs cycle plus two unique enzymes, malate synthase and isocitrate lyase, is one and only our glyoxylate cycle. Glycolate pathway, Kelvin cycle, TCA cycle is not applicable here. Our correct answer is glyoxylate cycle. For these, I would suggest you all the 4 cycles just revise once again so that it remains fresh in your mind. Next question, the maximum coiling of the chromosomes is observed during packaging of meiosis. No. Metaphase of mitosis, absolutely correct. Maximum coiling occurs, that is why they look dark colored, short and condensed. And then anaphase of meiosis, no. Telophase of meiosis too, no. So, our correct answer is metaphase of mitosis. Next, during replication, the long strands of DNA in a cell's nucleus get tangled, making it harder for a cell to read genes. But dash enzyme can prevent tangling. DNA topoisomerase is the correct answer. Ligase will join 2 fragments. Replicase is not applicable here. Isomerase is not applicable. The correct answer is DNA topoisomerase. Next, which of the following are the non-coding RNAs? rRNA, microRNA, messenger RNA is option A. Option B, rRNA, tRNA, mRNA. Option C, miRNA, tRNA, mRNA. And last option, rRNA, miRNA and tRNA. See, wherever mRNA is present, how can we mark that to be our correct answer? Right, so they will choose an option where mRNA is not there. So, rRNA, miRNA and tRNA, that is option D is your right answer. Next question, in the final step of protein synthesis, the folding of many proteins is made more efficient by a special class of protein called Gag-Paul fusion protein. Has no role here. Protease, that is what, protein degrading enzyme, has not got no role here. Ubiquitin has also got no role here. The only answer here is, the correct answer is chaperone. Next question, which of the following process takes place in 3' to 5' direction? This is a very important and very interesting question. We all know that DNA replication, RNA replication and RNA splicing takes place in 5' to 3' direction and not 3' to 5' direction. The only thing that takes place in 3' to 5' direction is RNA editing. So correct option is option A. Next, in eukaryotic cells, the tRNA genes 5S are RNA genes, and some snRNA genes and genes for other small RNAs are transcribed by RNA polymerase 3. RNA polymerase 1, 2 and reverse transcriptase has no role here. Our correct option is option C. Next question. The role of gRNA is, gRNA stands for guide RNA. Its role is RNA, it has an important role in RNA editing, not in polyadenylation, cell splicing and chemical modification of rRNA. It plays an important role in RNA editing. Coming to the next question. DNA repair pathway that can repair the damage caused by large change in the structure of the DNA double helix is called nucleotide excision repair. Base excision repair, mismatch repair and DNA interstrand cross-link repair is not associated with this. Nucleotide excision repair or NER can repair the damage caused by large change in the structure of the DNA double helix. So our correct answer is option D. Fermentation, next question. Fermentation of one molecule of glucose to ethanol and CO2 require dash number of oxygen molecules. How can we say that it requires oxygen molecule? Use common sense, you will get your answer. 0 oxygen molecules. Option C is your correct answer. Which of the following signaling molecule does not bind to nuclear receptor? It is written does not bind. So, cortisol, acetylcholine, testosterone, vitamin D3. The correct answer is acetylcholine. Next, which of the following describes cell-to-cell junction? This is again a very important topic. Please read all the possible cell-to-cell junctions that are there. First is occluding and claudine mediate cell contact. Option B, adherence junction occur as continuous bands. Option C, occluding junctions are abundant in cell under stress. Option D, inorganic ions are able to pass through anchoring junction to permit cell coupling. And correct answer is option B. Adherence junctions occur as continuous bands. Coming to the next question, gram-negative bacteria produces which of the following as their signaling molecule in quorum sensing? The one and only option is AHL, that is N-acyl homoserine lactones. Option B, C and D doesn't stand here. They do not stand to be the correct one. The correct answer is AHL. Which of the following is the tumor suppressor gene? P53, one and only P53 is a tumor suppressor gene. Question number 30, the next question. Rous sarcoma virus was first discovered in chicken, not in monkey, rat and rabbit, but it's chicken. This is something very important you should know. Where are they discovered? We will discuss the rest of the questions in our next lecture. Till then, stay tuned. We are soon going to meet with our part 2 or second part of our question paper discussion. Till then, thank you. All the best for your exam and happy learning. Bye-bye.